So hello everyone, welcome back. And guys, today we are starting with this session which is very relevant and very important for examination, because from today itself we are starting that part which we were waiting for. And what is it? So we are going to study theory first for the first 3 days, and on the fourth day we are going to have the most expected questions plus the previous year questions, which will make our examination very easy, and obviously we'll be clearing it in the first attempt. So yes, we have completed 3 topics of physics in the last week, and on the Friday, we had that session in which we had most expected questions plus previous year paper. Now, today we are going to have the discussion on the cell topic, which is the first chapter. Second chapter is related to the nutrients, and the third part will be related to the heart. So, today is Tuesday, Wednesday will be nutrients, Thursday will be heart, and on the Friday, the most expected, the previous year papers, everything will be sorted on Friday, because I am preparing free ebooks every week for you and they'll be delivered to you. How we can do that? So we'll be discussing that in the last part of this chapter. But as of now, let's start with the topic. And what's the first topic? The topic is cell. And what is cell? So cell is basically the unit of life, or we can say the structure of life. Why we are saying that cell is the unit of life or the functional unit of life? So there's reason behind it. What is it? We were born because of these cells only. Yes. So right from our childhood, these cells have played a crucial role in building up our body, in the repairment, and obviously in the cell division. So let's have a look. So guys, first of all, here the introduction about the cell. All the living beings, whether it is squirrel, whether it is tortoise, mushroom, bacteria, snake, whatsoever comes in your mind, all the living beings are made up of this cell only. So cell is the basic structure and functional unit of life. So that's what it is mentioned here. So a cell is the fundamental structural and functional unit of all the living beings, because as we know that when we were born, the sperm cell that was received from father and the egg cell that we have got from mother, when they fused together, they formed zygote, and that zygote, it was implanted in the uterus portion of our mother, and there we were the ones who took 9 months to develop the whole body, and the first organ that was developed was our heart. All these questions were asked in the examination, so let's go with the discovery first. So Robert Hooke was the one who discovered this cell in the year 1665, but this was the dead cell. What about living cell? So living cell was discovered by Anton von Leeuwenhoek, and he was the first one to describe a live cell, and he found this live cell in the pond water. What about Robert Hooke? So he discovered this cell in the cork, and that was the question which was asked previously, where it was found. So he observed the dead cells in the cork, which is a wooden structure, and when he observed there using the simple microscope, he found that there are some room-like structures. He felt that it looks like it's a honeycomb structure, and in the honeycomb, if I look at these parts, they look like rooms, right? And what is the synonym of room? That's cell. So from here, the cell actually term came in. Understood? See, it was as simple as that. Now proceeding further, this was just a discovery, but who actually gave the cell theory? That is, all the detailing about the cell was done by whom? So there were two important people. One was Matthias Schildine and second one was Theodor Schwann. Let's move forward with that. So yes, talking about the cell, the invention of the compound and electron microscope revealed the structural details of the cells. So obviously, if I need to go into the detail, I need some devices like these. And yes, here, if I talk about cell theory, so Matthias Schildine and Theodor Schwann, they were the one who gave this cell theory. And according to them, so who was Matthias Schildine? So he observed plants and he told that they are composed of cells. So that year was 1838, and Theodor Schwann in next year, that is in 1839, reported that the cells had a tiny outer layer of plasma membrane. He also found that plant cells have cell wall. So yes, he proposed a hypothesis that animals and plants are composed of cells and are basically the products of the cells. So this was in the starting, but later on there were more detailing of the cells done. And who was here? So here we had Rudolf Virchow, which actually gave the extended part of the cell theory. So what all points were given by Rudolf? So let's have a look. So Rudolf was the one who in 1855 explained that cells divided and the new cells are formed from pre-extinct cells. So he modified the cell theory, and the important points were all living beings are composed of cells and are products of cells, and also all the cells arise from the pre-existing cells. But that's not enough. There's one more important point. That is, cell theory is applicable for what? So obviously cell theory is applicable for us, that is we are animals, right? So humans, cats, dogs, snakes, for everything. Also it's applicable for bacteria and all, but please remember, cell theory is not applicable for virus, and that was again from your previous year paper. You must remember this, and that's very important. And wherever you feel like, sir, I have this doubt, please ask me, because I'm taking these sessions in a way that most of the things that are important for the examination will be covered here. And do not worry what kind of questions are framed, I'm already discussing it along with the theoretical part. But in order to check your knowledge, Friday is welcome for you. So in the Friday part, we'll be discussing about this, then heart, and also the nutrients part, where we'll discuss the questions. Okay, proceeding further. Now let's go with the overview of the cell. So cell contains what? So there's an outermost part that is known as cell membrane, right? That's also known as plasma membrane. Then there is the liquid which is present there, that is known as cytoplasm. Then apart from that, there's a nucleus which is there. So cytoplasm is a semi-fluid matrix where cellular activities and chemical reactions occur. So this keeps the cell in the living state. Then what's the next part? Ribosomes, they are non-membrane bound organelles which is found in the cytoplasm, chloroplast, mitochondria, and on rough endoplasmic reticulum. And also remember, these ribosomes are the ones that are known as protein factories. Why? Because these ribosomes are the ones that are involved in the protein synthesis. So obviously, they are known as protein factories. So these are one-liners which you must remember. Protein factory, that is ribosome. Then mitochondria, what is that? Powers of the cell. Lysosomes, what are they? They are known as suicidal packets. Chloroplast, they are known as kitchen of the cell. So these are one-liner questions which are regularly and repeatedly asked in the examination. Fine. So that's done. Proceeding further. So this is the cytoplasm, that is the liquid portion which is present in the cell. This bluish color is representing that thick structure that's present in the cell, and all the cell organelles are actually present in this part only. Then if I talk about this ribosome, so these ribosomes are found freely floating in the cytoplasm and also on the rough part of the endoplasmic reticulum. So that's involved in the protein synthesis, and that's why it is known as protein factory. Then if I'll talk about the cells, so cells are of two different types. Either they'll be fully developed or they will be undeveloped. So if cells are fully developed, just we can take our example, or you can take the example of plants. So the animals or the plants, these cells are fully developed, and hence we can say that they are what? They are eukaryotic cells. And the cells which are undeveloped, the perfect example will be bacteria, and they are known as prokaryotic cells. Fine. Okay. So see, if I talk about the smallest cell, that is mycoplasma, that is again undeveloped. So that's the example of prokaryotic cell. But yes, the smallest cell is the mycoplasma, the longest cell is the nerve cell, and the largest cell is the ostrich egg. What about human body? So within the human body, the largest cell is the female ovum, the smallest cell is the male sperm. Okay. So here, largest isolated single cell, that is the egg of the ostrich, and the longest cells are the nerve cells, as already discussed. What is the size of bacteria? So that's 3 to 5 micrometer, and human RBCs, they are 7 micrometer in diameter. So these are the images which is going to give you the clarity about the largest cell, that is the ostrich egg or the nerve cell. These are the bacteria, and according to their shapes, their naming is done. I'll teach you about it in the animals and plant kingdoms, that yes, according to the shape, bacteria have different names like bacillus, spirillum, and so on. Okay, further. So these are the RBCs which are biconcave in shape, and their number is 5.5 millions within our body. Yeah, that's helpful in the transportation of the oxygen. So now, if I'll talk about the plants, so plants have different kind of tissues, and humans or animals have different kind of tissues. So these are the tissues which are present in the animals, and these are the tissues of the plant cell. As discussed, cells are of two different types. They may be prokaryotic or eukaryotic. Depends. It's fully developed eukaryotic or undeveloped prokaryotic. So here kingdom Monera comes in, where the example can be taken as bacteria. What about eukaryotic cells? So these are the cells which are fully developed. Fine, cool. Next. So here, if I talk about these prokaryotic cells, so these are the cells which are undeveloped, which are not fully developed. And here, this is the image of bacteria, right? So see, if I talk about prokaryotic cells, so these are the cells where no membrane found. So they have no membrane bound, sorry. So there is no membrane bound nucleus and organelles. So they include bacteria, blue-green algae, mycoplasma, PPLO, that is pleuropneumonia-like organism, that is double PLO. Then these are generally smaller and multiply more rapidly than eukaryotic cells. So it was asked in the examination previously. There were 4 options given and it was asked which of the following organism reproduces in the fastest mode, and their option was bacteria. So yes, these type of questions are framed in the examination. So here they vary in the shape and size. So just see, these are 4 type of bacteria only. So these are also the bacteria, but they are rod-like structures. They are actually spherical in shape. These are cyanobacteria and these are mycoplasma or PPLOs. Okay. So bacteria have 4 basic shapes and you need to remember these names as they are asked in the examinations repeatedly. So if they are bacillus, they will be rod-like structures. If they are spherical, they will be known as coccus. If they are comma in shape, they are known as vibrio, and spiral in shape, so they are spirillum. I'll show the image also. So here you have bacillus. So then the second one is coccus. So just you can see they have a sphere shaped structure. Then we have vibrio, this comma shaped structure, and spirillum. So you can link it with this ring, right? So spirillum, vibrio, coccus, and bacillus. So these are the 4 types of bacteria, and I guess this part is clear now. So let's proceed further. Okay. So if I talk about the cell organelles of the bacteria, that is, I'm talking about the prokaryotic cells. So what are they? So they have cell envelope, mesosomes, and chromatophores. Then they have nucleoid, they have flagella, they have pili and fimbriae, they have ribosomes, and yes, there are other inclusion bodies. So guys, here the one thing that is common in the animal cell and here is ribosome. Ribosomes are found at both the locations. Fine. So let's proceed further. Okay. So guys, it's the time to understand the eukaryotic cells now. So if I talk about the eukaryotic cells, so eukaryotic cells are the ones which are fully developed, right? So here, yeah. So it's the time to understand eukaryotic cells now. So what are eukaryotic cells? So these cells are fully developed. Okay. See, so you can talk about the protista, you can talk about the fungi, you can talk about the plant, or you can talk about the animal. So only kingdom Monera had prokaryotic cells. Rest all the kingdoms will have eukaryotic cell only, and that means the cell which is fully developed. Let's have discussion on more about it. So I guess one of the student has doubt. So is there any schedule for these classes? Yes, we can. The timing is fixed. You will be having these classes daily at 5 p.m. Sharp at 5 we'll start, and we are starting with the cell today. Tomorrow we'll be discussing the nutrients chapter. Day after tomorrow we'll be proceeding with the heart chapter, and on the Friday at the same time, that is all these classes will be there at 5 p.m. sharp. So it's from Tuesday to Friday. So you can just join for these first 3 days for the theoretical portion and for the most expected questions and the previous year papers on Friday. That means Tuesday to Friday sharp at 5. Fine, done. Okay, so yes, these were the 4 different kingdoms. We'll be discussing about it in detail in the animal and plant kingdoms part, that is in the last part of the chapter. But as of now, they have well organized membrane bound nucleus and organelles, so they are fully developed. Also, presence of membranes gives the clear compartmentalization of the cytoplasm. They have complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. So yes, chromosome number is not same in all the organelles. If I talk about our body, so that's 46 in number. Fine. Next. So I'm just talking about mine part. So I am taking the science section here. So yes, for the science section, you can join at 5 and all the classes have their own different timing. Fine. Okay, so talking about the cell organelles in this nucleus, just have a look. Sorry, in this prokaryotic cell. So this is the structure of the cell. Now here you'll find cell membrane, cell wall, endoplasmic reticulum, Golgi apparatus, mitochondria, plastids, ribosome, cytoskeletal, cilia, flagella, centrosome, and centrioles, nucleus, microbodies. They all are present here. Now there's some difference in the animal and plant cell. What is it? So if I talk about the cell wall, so that outermost layer of the plant cell, that's known as cell wall. So that's only there in the plant cell only. Similarly, chloroplast, that's only the part of plant cell. But in the animal cell, is there any different thing which is not there in the plant cell? Yeah, there is. Like you can talk about cilia. So they are only present in the animal cell. Okay. Now, first of all, what are cell membranes? So cell membranes are the outermost part of the cell in the animal cell. Is it present in the plant cell? Yeah, obviously. It's also there in the plant cell, but that's not the outermost part, because in the plant cell we have one more layer, and that's known as cell wall. Okay, so just have a look. So this is the structure of cell membrane. So here we have lipid bilayer which is present. Here we have this cholesterol, sugar, proteins. So chemical studies on human RBC show that cell membrane is composed of lipid bilayer, protein and carbohydrate. This was the question from SSC JE and also from SSC CGL. Then lipids, mainly the phosphoglycerides, have outer polar head and inner hydrophobic tails. So the non-polar tail of the saturated hydrocarbons is protected from the aqueous environment. Right. Okay. Now more about it. So ratio of the protein and lipid varies in different cells. So if I talk about human RBCs, membrane has 52% protein and 40% of the lipids. So it's asked in the examination, these cell membranes are made up of what? So the cell membrane which we people have, it's made up of lipids and proteins. Right. Proceeding further. So guys, based on the ease of extraction, membrane proteins are of two different types. It may be integral or it may be peripheral. Fine. So that's done. Now if I talk about the fluid mosaic model of the cell membrane, so it was given by Singer and Nicholson. Only this part is important. Don't need to go in the depth here. Then the next part, so that's from the yeah. So now talking about this cell wall. Cell wall is only and only present in the plant cell. It's not there in the animal cell. So the outermost layer, this is what cell wall is. So cell wall is made up of what? So that's made up of cellulose. So it may be cellulose, hemicellulose, pectin. So it is a non-living rigid structure which is found on the outer to the plasma membrane of the fungi and plants. See, cell membrane or plasma membrane is present in all the categories of eukaryotic cells. We as an animal, we do not have any cell wall. In the plant cell and in the fungi, they have cell wall. Both of them have. So that means just before the cell wall, they might be having cell membrane. That's what it is mentioned here. What is it written? It is a non-living structure found outer to the plasma membrane of the fungi and the plants. Cell wall of algae is made up of cellulose, galactans, mannanas, and minerals like calcium carbonate. Yeah, this part was asked in the examination previously. Okay, then in other plants, it consists of cellulose, hemicellulose, pectins, and proteins. So I am giving you hint here. Guys, I am going to discuss the same question because it was asked previously in the CGL and also in the SSC. Also, it was asked in CPO, this last line. So I'll be keeping this question in the Friday section for sure. And yes, one more thing, ebook will be ready by tomorrow. So I'll just ask the team to just go for the quality check, and you will be getting this free ebook after the session on Friday. And it will be done every Friday basically. Okay. So guys, cell wall of a young plant cell is capable of growth, so it gradually diminishes as the cell matures and secondary wall is formed on the inner side towards the membrane. Okay, so this part is sufficient for this. So what is the function of cell wall? Let's have a look at that. First of all, it is going to give the shape to the cell. It is going to give the protection from mechanical damage and infection. Then it is also, as discussed, it is also giving the support, right? So it helps in the cell to cell interaction, and it also acts as a barrier to the undesirable macromolecules. In short, it is also giving the safety. That's important. Fine. Now it's about the endomembrane system, which includes endoplasmic reticulum, Golgi bodies, it is Golgi apparatus. So here, let's have a discussion on them. So they include endoplasmic reticulum. They'll also include the ribosomes because they are attached to it. So Golgi complex, lysosomes, vacuoles, they all are under this endomembrane system. One by one, let's discuss. So what is this endoplasmic reticulum? This structure. So this structure has two main parts. One is smooth, another one is rough. So here, this is rough endoplasmic reticulum. In short, it's mentioned RER. And this is the smooth endoplasmic reticulum, that's mentioned SER. So what's the difference between two? Rough endoplasmic reticulum includes ribosomes. Looks quite typical, right? Arey, I am there. Why do you feel it's quite typical? Just give me 1 minute. Just allow me a minute. So guys, if I talk about R, So from R, rough part, and from R, you can link it with the ribosome. So where this ribosome is found? This ribosome is found on the rough part of the endoplasmic reticulum, and ribosome was known as the protein factory. Why? Because it was linked with the protein synthesis. So this was again asked in this SSC that what is the function of this endoplasmic reticulum? So that helps in the production of lipid and proteins. So proteins are generated from rough endoplasmic reticulum and lipids from the smooth endoplasmic reticulum. That's it. So lipids are generated from smooth endoplasmic reticulum. I guess this part is sorted, and I don't think so that you are going to have any problem here, right? Perfect. Okay, so let me proceed further. So endoplasmic reticulum divides the intracellular space into two compartments, that is the luminal, that is inside endoplasmic reticulum, and extraluminal, that is cytoplasm. So endoplasmic reticulum also has two types, rough and smooth. So here the rough endoplasmic reticulum, they bear ribosomes as discussed on the surface, and they are involved in the protein synthesis and secretion. Also, smooth endoplasmic reticulum, these are the major sites for the synthesis of lipid. So these are important one-liners, and obviously all the questions will come from this part only. So please listen to it carefully, and wherever you feel like you have any doubts, just let me know. Okay. So now let's talk about Golgi apparatus. So guys, this is the structure of Golgi apparatus. This is the cis phase, this is the trans phase, and it is helping in the intracellular transportation. What it is doing? It's helping in the intracellular transportation. So just have a look. So it's densely stained reticular structures near nucleus. Firstly, it was observed by Camillo Golgi. So Camillo Golgi was the one who discovered it. So from this Golgi name, this Golgi apparatus is derived. Then they consist of flat disc sacs, that is cisternae, of approx 0.5 to 1 micrometer diameter. They are stacked parallel to each other. Cisternae are concentrically arranged with the convex cis, that is forming phase, and concave trans, that is maturing phase. Cis and trans phases are totally different, but they are interconnected with each other. Okay, so what are the functions? So they are helping in the intracellular transportation as discussed. They are also involved in the protein synthesis. So protein synthesized by the ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus. So it's asked where modification of the proteins are done. That's again linked with the previous paper. So the modification of this protein is actually done in this Golgi apparatus only. So they have two main functions. The first one is intracellular transportation, and the second one is protein modification. Fine. Okay, so this is also done. Next, so they are also helping in the formation of glycoproteins and glycolipids. Then lysosomes. So these are the lysosomes, which are also known as the suicidal packets. Why? Because it has enzymes, and if cell doesn't function properly, it bursts. And because of its bursting, what happens? It destroys the complete cell, and that's why it is known as the suicidal packet of the cell. Okay, so membrane bound vesicular structures formed by process of packaging in the Golgi apparatus. So lysosomal vesicles contain almost all the types of hydrolytic enzymes, and they are active at acidic pH. So they digest carbohydrates, proteins, lipids, and nucleic acids. Okay, talking about vacuoles, guys, you might be wondering why I'm using this type of image now of the cell. So guys, this image which is present in front of you, what is it? That's the perfect example of the plant cell. So within the plant cell, this central vacuole is present, but it doesn't mean that this vacuole is absent in the plant cell. This cell is present in the plant cell, but in the animal cells also they are present, and the only difference is the size of this cell is actually smaller in the animal cell. Fine. Okay, so vacuoles are found in both the parts. So these are membrane bound space found in the cytoplasm. They are linked with the storage and all. So they contain water, sap, exudative products, and other materials that are not useful for the cell. Vacuoles is bound by a single membrane that is known as tonoplast. And yes, it's present in the both animal and plant cells. So in plant cells, the vacuoles can occupy up to 90% of the volume of the cell. And here, these structures, they are actually the vacuoles, these blue structures. So in the plants, tonoplast facilitates the transport of ions and other materials against the concentration gradients into the vacuole. So yes, their concentration is higher in the vacuole than in cytoplasm. In amoeba, contractile vacuole helps for the excretion. In many cells like protists, food vacuoles are formed by engulfing the food particles. So that was about vacuoles. Mitochondria. Mitochondria are known as the powers of the cell. Why? Because they're involved in the protein, this energies part. Now, energy is produced in the form of what? That's stored in the form of ATP. And what is ATP? That's adenosine triphosphate. So if I'll go with the aerobic respiration, which takes place in the mitochondria, it gives you approx 36 to 38 ATPs of the energy. But if this respiration is anaerobic in nature, that is done in cytoplasm, and that time only 2 ATPs of energy is generated. So yes, since energy is generated, they are known as the powers of the cells. So they are clearly visible when strained. Their number, shape and size per cell are variable depending on the physiological activity. Okay, so here it is a double membrane bound structure, and that's involved in the production of the energy. That's it. Proceeding further, they were also known as the powers of the cell. Talking about plastids, that's the final portion of our chapter. So if I talk about these plastids, so these plastids are present in the plant cell. They are not there in the animal cell. So yes, here we have chloroplast, chromoplast and leucoplast. So what they are doing, let's see. So they are found in all the plant cells and in euglenoids. They are large sized, easily observable under the microscope. They consist of some pigments, and here they are chromoplast, chloroplast and leucoplast. Chloroplasts are involved in the green color, transferring. Chromoplasts are involved in different colors of fruits and flowers, right? And these leucoplasts are either white or they are colorless plastids. Okay, so they are chloroplast, chromoplast or leucoplast. So if I talk about chloroplast, so they contain chlorophyll and carotenoid pigments. So they trap the light energy for the photosynthesis. So yes, that's why it is known as the kitchen of the cell. Then chromoplasts, they contain fat-soluble carotenoid pigments like carotene, xanthophylls, etc. They give yellow, orange or red color to the leaves and to the flowers. And leucoplasts, they are colorless plastids of varied shapes and sizes with the stored nutrients. So they include amyloplast, that stores the starch, for example potato. They include Elioplast, that it stores the oils and the fats, and it also has aleuroplast that stores the proteins. So here finally, if I talk about this chloroplast, so they are double membrane bound organelles which is mainly found in the mesophyll cells of the leaves. They are lens-shaped, oval, spherical, discoid and ribbon-like organelles. This is the length and this is the width. And in short, yes, their number varies from 1 to 20 to 40, depends from cell to cell. So that's all from the cell chapter. What kind of questions are asked, I've already discussed about that in between the theoretical portion. In the next class, that is tomorrow sharp at 5, we are going to start the nutrients part, and day after tomorrow we'll be discussing the heart. So on the combination of these three chapters, we'll be discussing uh, PYQs and most expected questions on the Fridays. And guys, please be ready for this Friday session because your ebook will be ready by today's evening, and tomorrow I'll be sharing it with the team to go with the final check, and your ebook will be ready and you need not pay anything. I'm just giving everything for free of cost, including the session as well. So just let me know in the comment section how was the class, and also there's a homework question for you all, and it's very basic. Please tell me which of the following is known as kitchen of the cell. Option number A, mitochondria. Option number B, vacuole. Option number 3, chloroplast. Or option number D, it's Golgi apparatus. Let me see who is going to give the correct answer, and I'll personally reward you there. So that's all from my end. And also guys, if you want to take any of the paid batches, what you can do, you can just go to the Google, and here you will type Oliveboard. Once you tap Oliveboard here, you will click on the first link, and here on the dashboard, you will find SSC plus Railway Super Elite batch, which includes both Hindi and English medium courses, where you will have tests, live classes, recorded classes, everything is here. 1200 plus live sessions of SSC, 600 plus live session of Railways, 300 plus concept videos, 900 plus mock tests, type and tests, everything is here. You just need to click on the enroll now option. And since we are celebrating Father's Day, so you have a coupon code DAD that is going to give you the maximum discount of up to 59% off. So you will just click here, type the code DAD, that is for father, and you will get the maximum discount. So that's all from my end. So we'll meet tomorrow. And now tomorrow's topic is nutrients. So do not forget to join tomorrow at sharp at 5. So take care guys. Bye bye. Jai Hind. And for the examination, all the very best. Please take care guys.